So this is the second experiment that we did today. So it is on separation of amino acids by paper chromatography. Now chromatography, it is a method for separating the molecules. Okay, you can separate molecules using chromatography. So in this particular experiment, we are going to use amino acids. You can even separate different molecules such as pigments. Okay, so you can separate. Now in this method we use a moving solvent. Okay, so let me just show you. We have already prepared a solvent here. This is the glass jar that we are going to keep our paper inside, okay, the chromatography paper, and this is the solvent that we are going to use in this experiment. Now, the principle here is that we are using a moving solvent. This solvent, it will move in a chromatography paper, and we need that chromatography paper. It acts as a solid phase, okay, as a support. Now the amino acid sample that we are using, okay, mixture of amino acid, you have to load in this paper. This is a specialized paper for this experiment. It is a chromatography paper, okay. It is made up of cellulose. So cellulose is the constituent of this chromatography sheet, which is suitable support, okay, which acts as a support, and where water is being absorbed in this paper between the cellulose fibers and form a stationary hydrophilic phase. So using this paper chromatography, we will load our sample here, and that sample will move along with the solvent. So here the solvent is a moving solvent. It is a mobile phase, and this chromatography, it acts as a support phase, as a solid phase. Okay, so the molecule in the sample, they will be retained based on the functions of the molecular size, the shape, the binding affinity to this solid phase or the paper. And here, like I said, this paper chromatography can be used to separate different molecules, okay, separating different molecules. So here we are going to separate the amino acids, and also we will calculate the RF value, that is the ratio between the distance which is traveled by the solvent, okay, to the distance traveled by the amino acid, because we are going to load the amino acid here. So the distance traveled by the amino acid to the distance traveled by the solvent. So in that way we are going to calculate the RF value. Now first thing, that this is the glass jar that we require in this experiment, okay. It has a cover also, and we need the small capillary tubes for loading the sample in the chromatography paper. As you can see here, these are known as the capillary tube. Using these capillary tubes, you can add the sample in the paper, and this is a mixture of 2 amino acids that I have already prepared. And after loading the sample in the paper, see, I will just show you this one. What we have to do first of all, when you get a chromatography paper, you will load down this, you will take a diameter, you will take this origin from the edge of the chromatography paper, a distance of 2.6 centimeter. Okay, so you will mark that as the origin. Okay, so you will take from here till here 2.6 centimeter, mark this as the origin, and you mark a point where you will load your sample. Okay, so you mark a point where you will load your sample. Then with this, using this capillary tube, you will take your sample and you drop it in this point, okay, in this origin. Then you dry. Again, you load your sample, you dry, you try to load your sample 4 to 5 times. Okay, so after loading the samples, this paper, we are going to keep it in the jar, in this jar which contains a solvent. So while loading the paper here inside, the bottom of the paper should touch the solvent, okay, in order to allow the solvent to pass through this chromatography paper. So the bottom of the paper should just touch the solvent, but it should not touch the glass jar. Okay, it should not touch the walls of the jar. It should you should hang it in a thread. You can see here there is a thread. We will hang this paper in this thread, and the bottom of the paper will touch the solvent, and we will allow the solvent to pass through this solid phase. So when you load your sample, what happens when you keep it inside the solvent? These samples, the amino acid sample, they will move along the solvent. So after some time, when the solvent run, okay, when the solvent, it has moved about 3/4 of this paper, you will take out this paper, you dry it, and in order so that these amino acids will be visible, we will add ninhydrin. Again, the first reaction using ninhydrin, okay, to detect amino acids. So here also, since these are amino acid sample, we will use ninhydrin. We will spray ninhydrin in this paper. Then these different color samples are visible here. These are amino acids. Okay, you can see two amino or three actually. One, this is more darker and this is more lighter in color. So it is glycine and tryptophan. So you see, based on the RF value of the amino acid, okay, it will move that much. So tryptophan, it is moving more farther as compared to glycine. So in order to calculate the RF value, we will we'll measure the solvent front. Okay, you see here, for example, it is 8.7 centimeter. Then tryptophan, it has moved 4.2 centimeter. Then glycine, it has moved 2.1 centimeter. So the RF value can be calculated from here. The RF value of tryptophan, it will be 4.2, that is distance traveled by tryptophan, divided by the distance traveled by the solvent, okay, by the solvent front. Now you can see here the calculation. The RF value of tryptophan, it is 4.2 by 8.7, which is 0.48 centimeter, and the RF value is less as compared to tryptophan. You can clearly see here that these two amino acids, they get separated according to their RF value, okay, because of their different molecular weight. So they will, the amino acid with high molecular weight, it will move slowly, whereas the one with less molecular weight, it will move faster than the other. So what we can conclude from this experiment is that different amino acids, you will see that they have different RF values comparing with the known amino acids. So suppose I have a mixture of unknown amino acids. So by loading these unknown amino acids with the known amino acids, okay, that have the same RF value. So suppose I have the unknown amino acids also, I load here the unknown. Okay, suppose I load in this point the unknown amino acid. So suppose that amino acid, it has the RF value like tryptophan. So then from that we can conclude that the unknown is tryptophan. So in this way you can, unknown amino acids, they can be found out from the RF value. And these different amino acids, they can be separated by paper chromatography, okay, by this experiment.